In the inner ear of zebrafish, there are 3 pairs of sac-like end organs: the utricle, the saccule, and the legana. The utricle locates most anteriorly, while the saccule and the legana closely attach to each other and locate posteriorly to the utricle. After gross dissection, the saccule and legana stay attached, and the stone-like structure, otolith, can be easily seen in each end organ. In the following fine dissection, the saccule and legana are separated from each other by cutting from the ventral side. Otoliths are removed and the non-sensory epithelial tissues are trimmed off to get sensory epithelia tissues from all 3 end organs.
Hi, I'm Jean Liang from the lab of Sean Burgess in the Developmental Genomics section of the National Human Genome Research Institute. Today, we'll show you a procedure for the fine and gross dissection of the inner ear sensory epithelia from the adult zebrafish. We use this procedure in our lab to study the gene expression in the inner ear sensory epithelia. So, let's get started.
To begin the dissection, euthanize the adult zebrafish with buffered MS-222.03%. Decapitate the fish. Then cut off the lower jaw. Next, place the head on the dissection pad with the ventral side up and immobilize the tissue with insect pins. Under the dissection scope, remove the soft tissue ventral to the skull with fine tweezers. Using the same tweezers, crack open the ventral part of the skull capsule. Finally, fix the fish head in a small plastic container in 4% paraformaldehyde and 1x PBS at 4 degrees Celsius overnight. The following day, discard the paraformaldehyde. Because paraformaldehyde is considered hazardous waste, please remember to dispose the used paraformaldehyde solution according to the lab safety regulations of your institution. And rinse the fixed fish head in 1x PBS 3 times, 5 minutes each time. Now that the fish head is fixed, gross dissection of the inner ear can begin. Start by placing the fish head on the dissection pad with the ventral side up. Immobilize it with insect pins. From this point forward, the rest of the work will be done under the dissection scope. With a pair of fine tweezers, remove the skull bone over the inner ear and then carefully pull out the inner ear tissues, the utricle, the saccule, and the leganae. The saccule and legana are tightly attached to each other and thus pull out together, while the utricle, anterior to the saccule and legana, usually detaches from the other end organs during dissection. Last, put the inner ear tissues in 1x PBS. Following the gross dissection, one can proceed with fine dissection of the inner ear sensory epithelia. The fine dissection is carried out on a clean glass slide in a drop of 1x PBS. Use 2 pairs of fine tweezers to remove the utricular otolith from the utricle. With a pair of fine tweezers and a needle blade, trim off the non-sensory epithelial tissue and the innervation from the epithelia. Place the saccule and legana, which are still attached to each other, in the PBS drop with the ventral side up. Next, remove the leganar otolith. Use the needle blade to carefully cut in between the saccule and legana. Finish by trimming off the excessive non-sensory epithelial tissue in the innervation with fine tweezers and a needle blade. The sensory epithelia are now ready for staining. To begin the staining, rinse the sensory epithelia with PBT for 3 times at 5 minutes each time. Next, incubate the epithelia with Alexa Fluor 488 phalloidin at a 1 to 1000 dilution for 30 minutes at room temperature. The phalloidin binds to actin filaments in the cells. As the apical bundle of the hair cell in the sensory epithelia is an actin-rich structure, it will be strongly stained by the phalloidin. Rinse the sensory epithelia with PBT for 3 times at 10 minutes each time. To view the cells, mount the epithelia onto a slide with VectaShield hard-set mounting medium with DAPI. Check the stained tissue under the fluorescent microscope with FITC filters. After successful dissection and staining, intact epithelia with strong hair cell bundle staining can be seen under the microscope. Under lower magnification on the fluorescent microscope, the most distinguishable structures are the hair cell bundles, each of which appears as a bright dot in the sensory epithelia. It is the most strongly stained structure because it is very rich in actin. The bundle is a unique apical structure of the hair cell that can be used to easily identify hair cells, as well as identify the regions of the sensory epithelia.
We've just shown you how to dissect the inner ear sensory epithelia from the adult zebrafish. If you skip the step of fixation, the procedure can also be used for preparing tissues for the extraction of DNA, RNA, and proteins. So that's it. Thank you for watching and good luck with your experiment.